Yo, it's Dr. James Utley, your resident code breaker, data scraper, and I'm here to take you on a journey of what's happening with the old Syndicate Laboratories. So you can find this at syndicate-lab.io, syndicate-lab.io. All right, there's like really three main projects. We're going to talk about the first one right now, which is BioCypher. So we're going to head over here to BioCypher. So if you have not heard of BioCypher, it's a biological cryptography or DNA cryptography open protocol that I've been developing so folks can use it to communicate via the mechanism using DNA embedded in things, right? So that's like plants, bacteria, living tissues, etc. And I spoke about it at DEF CON 33, and there's here. You can find it on the DEF CON YouTube channel, or you can just click it here, and and so we're going to go over these two documents here real quick, and that is the BioCypher Honk Manifesto, right? Every good project deserves a manifesto. And this one here is the technical guide and protocol specifications. And down below here is a little added bonus. That's the biohacking village at DEF CON. It's a separate presentation that kind of goes over what, DNA cryptography or biocryptography is, and, how to and basically goes over the project. All right. Oh, here's the last one is the GitHub repo. This is open source, so please contribute. All right, so let's begin. Let's begin first off with what we're going to call the digital panopticon. So if you know what the term panopticon is, it's basically we have created a digital prison. And so we have to, in the future, have a way to communicate amongst each other, probably over great distances, which means space travel, etc. And this is the manifesto of those that also share this opinion or not. I don't know. But it's a manifesto that kind of goes over the why it's important, and using biological material as a communication media and why that is important also. Okay. The next item here is the technical guide or protocol specification. So if you see that BioCypher, it's an open source application, and it encodes digital data to DNA sequences, right? So using DNA as a storage mechanism, a communication mechanism. And this right here is kind of the technical protocol specification for the application, which really has only three modes. The first one being DNA to binary, or sorry, basic binary to DNA. The second one being optimized for a decoding step, which is later in the line, which is using a nanopore sequencer. And it's all outlined here. And then the last one is adding a layer of traditional encryption. I don't know if you can say traditional, but AES 256 encryption. And the protocol goes over all of that, right? So the protocol, it goes over the architecture, which is like the application layer, the encoding layer, the DNA mapping layer, the physical layer, et cetera, et cetera. And it gives you all of the encoding algorithm is here, and the decoding algorithm is there, and so on and so forth. Nanopore mode specification, and how to best optimize for that. Okay, well, the last thing I wanted to show you around this was the GitHub. So this GitHub is public. Not a lot of peeps working on it except for me, but if you'd like to, it's all here. So check it out, and you can go over it. Throw it a star. Please help a guy out. Throw a star. And also, yeah, follow me on the GitHub for all the latest updates. All right. Well, now, so let's head back here. I would be remiss if I did not talk about the other projects of Syndicate Laboratories, which I'll give each individual video for each. But the next one here is a preview. This is nucleogenesis. It's a temporal framework for eukaryotic nuclear blueprint archival and reconstruction. So what that means is it's building from the bottom up, building a nuclear, meaning the nucleus, architecture that takes information, builds a framework of a nucleus, but then you could further give to a synthetic biology or a biologist or that type of person, that type of field of science, to then be able to reconstruct that nucleus. Super theoretical. It's kind of a collect now, decrypt later philosophy, and has a little video there. But I'll do a whole video on it. And then the last one is Stellar OS, which was actually provided me to be able to win the first round of the Humans in Space Challenge, and that is basically genetically engineering mesenchymal stem cells, or MSCs, to overexpress ESOD, extracellular superoxide dismutase, and one other marker, which you can read about in the paper. It's available. You can read it in the paper. It's right here. But anyway, that's what's happening for this year. in the old Syndicate world and Syndicate Laboratories. This, the science of code destruction and other synthetic delusions of the Syndicate. Okay. All right. My name is Dr. James Utley. I'll see you next time. Peace.